Welcome back to the Deep Dive. Today we are taking a deep plunge into one of the most, well, compelling and maybe frustrating areas of human health, autoimmune diseases.
Absolutely. It's this fundamental problem where the body's defense system, the immune system, gets its signals
Yeah,
crossed.
it turns inward, right?
Yeah.
It starts attacking our own healthy cells and tissues. It's like the ultimate internal security failure.
Precisely. Conceptually, it's a huge paradox in immunology, this idea of self-attack.
And it forces us to really grapple with how our bodies know what's us and what isn't.
It does. And while it sounds maybe like a niche scientific puzzle, the scale is actually enormous.
Right, the numbers are quite startling.
Yeah, we're talking about a common and actually growing public health issue. Current estimates suggest maybe 5 to 10% of the U.S. population is affected by a recognized autoimmune disorder.
5 to 10%. That is a massive number of people.
It really is.
And what's particularly concerning, as our sources highlight, is that the incidence rate seems to be climbing.
Especially in high-income, sort of westernized countries, it's a pattern we're seeing more and more.
Before we dive into why that might be happening, we really need to nail down what officially counts as an autoimmune disease. It's not as simple as just finding some autoantibodies, is it?
No, absolutely not. That's a critical distinction immunology makes, separating just biological noise from true disease, from pathology.
Because you can find these antibodies just floating around sometimes.
You can, yeah. Especially as people get older, it's quite common to detect autoantibodies. But their mere presence, that alone, isn't enough to diagnose an autoimmune disease.
Okay, so when does it cross that line? When does it become, a genuine pathologic disorder? What are the requirements? I think the source mentioned 3.
That's right. The sources lay out 3 key hurdles, essentially. First, there has to be clear evidence of an immune reaction. Could be T cells, could be antibodies that is specifically targeting a self-antigen or a particular self-tissue.
So step 1, identify the attacker and the target within the body.
Exactly. You need that specificity.
And the second requirement.
The second one is maybe the most crucial from a biological standpoint. There has to be solid evidence that this immune reaction is of primary pathogenic significance.
Meaning
Meaning the immune response itself is directly causing the tissue damage. It's not just showing up after the fact.
Okay. So you have to rule out situations where, say, a toxin or an infection damages tissue first, and the immune system is just cleaning up the mess.
Precisely. The immune attack has to be the root cause, not just a consequence of some other damage. The immune system is the aggressor here.
Got it. The aggressor, not the responder. And the third hurdle.
And finally, number 3. There must be an absence of another well-defined cause that could explain the whole clinical picture.
So rule out everything else, basically.
Pretty much. If you meet those 3 criteria, specific immune reaction, primary cause of damage, and no other explanation, then you can confidently label it a true autoimmune disorder.
Okay, that makes sense. So once we've established it is an autoimmune disease, we see it doesn't just look like one thing, it's a whole spectrum, right?
Right.
Depending on what's being attacked.
Exactly. The target really dictates the disease. We usually think about it on a spectrum from organ-specific to systemic.
Let's start with organ-specific. That sounds pretty straightforward.
It is. In these diseases, the immune response is really tightly focused, aimed at antigens that are found only in a single organ or maybe one type of tissue.
And the classic examples here would be things like type 1 diabetes or multiple sclerosis.
Perfect examples. In type 1 diabetes, the attack is incredibly specific. T cells and antibodies go after the insulin-producing beta cells in the pancreas. That's it.
Leading to insulin deficiency.
Right. And in multiple sclerosis, or MS, you have autoreactive T cells targeting myelin, which is that fatty sheath insulating nerve fibers, but specifically within the central nervous system. Very focused attack.
Okay, so that's one end. What about the other end, the systemic diseases?
Systemic diseases are where the immune system seems to lose that focus almost completely. It starts reacting against antigens that are widespread throughout the body.
So the damage isn't localized?
Not at all. You get generalized lesions, damage appearing in multiple organ systems pretty much simultaneously.
And the prime example here is lupus. Systemic lupus erythematosus.
Absolutely. SLE is the textbook example.
Huh.
The range of antibodies in SLE is just staggering, targeting things inside the cell nucleus like DNA, but also platelets, red blood cells, these protein phospholipid complexes.
And because those targets are basically everywhere.
The disease can manifest everywhere. Skin, joints, kidneys, the membranes lining organs. It's truly systemic breakdown.
And then the source material pointed out an interesting Middle ground case, good pasture syndrome. Where does that fit?
Yeah, good pastures is fascinating. It targets basement membranes, specifically a component of type IV collagen.
Okay. And where do you find that?
Critically, you find it in the filtering structures of the kidneys, the glomeruli, and also in the air sacs of the lungs, the alveoli.
Ah, so the antibody is specific, but the target molecule happens to be in two key places.
Exactly. So it's not systemic like lupus, where everything's a target, but it's broader than just one organ. The location of that specific antigen dictates the pattern of disease. It really highlights that molecular specificity.
Okay, that framework, organ-specific, systemic, and those in-between cases helps visualize the scope. But it all comes back to the big question. How does this tolerance, this self-recognition, fail in the first place?
Right. Because the default state, the normal state, is immunologic tolerance. That's the unresponsiveness to self-antigens that lets us, as the source puts it, live in harmony with our cells and tissues.
It's a carefully regulated system. How is it organized? How does it normally work?
Well, we generally divide the mechanisms into two main categories, sort of like two layers of security: central tolerance and peripheral tolerance.
Central tolerance.
Oh,
It's
yeah.
like basic training.
That's a perfect analogy. It happens during lymphocyte development in the primary lymphoid organs, the thymus for T cells, and the bone marrow for B cells. It's quality control before the cells are released into the body.
Let's focus on T cells in the thymus first. What's the main mechanism there?
For T cells, the primary process is negative selection, which is also called clonal deletion.
Sounds ominous.
It is for the T cell. If an immature T cell shows it binds too strongly to a self-antigen presented to it in the thymus, it's essentially flagged as dangerous.
And what happens then?
It's instructed to undergo apoptosis-programmed cell death. It gets eliminated before it can cause trouble.
But wait, how does the thymus, this central organ, know about all the specific proteins that are only found out in, say, the pancreas or the thyroid? How can it test for reactivity against those?
That's where this absolutely crucial protein called AIR comes in. AIR stands for autoimmune regulator.
Okay, what does AIR regulate?
AIR has this remarkable function. It turns on the expression of hundreds of these peripheral tissue-restricted antigens within the thymus itself, specifically in certain epithelial cells there.
Seriously. So antigens normally only found out in the body. Tissues are deliberately made inside the thymus.
Yes. It's like bringing samples from all over the body to the central training ground. This ensures that developing T cells are tested against a much wider repertoire of self-antigens than just those normally present in the thymus.
That's incredibly clever. It's like a comprehensive final exam before graduation.
It really is. And we know how critical it is because when AIR is defective, things go wrong.
There's a disease associated with that.
Yes. Rare individuals with loss-of-function mutations in the AIR gene develop a condition called autoimmune polyendocrine syndrome, or APS. They suffer from the destruction of multiple endocrine organs like the parathyroids and adrenals.
Wow. Direct proof of AIR's importance in preventing widespread autoimmunity.
Absolutely. It demonstrates how vital this central tolerance mechanism is.
Okay, so that's T cells being deleted if they're self-reactive. What about B cells developing in the bone marrow? Is it the same process?
It's a bit different for B cells. They have another option before deletion, a process called receptor editing.
Editing. They can change their receptor.
They can try. If a developing B cell makes an antibody receptor that binds strongly to a self-antigen in the bone marrow, it doesn't just get deleted immediately. Instead, it gets a signal to reactivate its gene rearrangement machinery.
The machinery that originally built the receptor?
Exactly. It tries to swap out the problematic light chain, essentially shuffling the genetic deck again to create a new receptor that hopefully is no longer self-reactive.
A genetic do-over. That's amazing.
It is, and our sources estimate it's quite common. Maybe 25% to 50% of developing B cells might go through receptor editing. It's a significant tolerance mechanism.
And if the editing fails? If the new receptor is still self-reactive?
Then the B cell gets the signal to undergo apoptosis. So deletion is still the backup, but editing provides a crucial second chance.
Okay, so central tolerance weeds out a lot of potentially dangerous cells through deletion and editing.
Yeah.
But it's not perfect, right? Some self-reactive lymphocytes must slip through.
That's absolutely correct. No biological system is 100% efficient. Some self-reactive T and B cells inevitably escape these central checkpoints and enter the circulation and peripheral tissues.
Which means we need that second layer of security you mentioned, peripheral tolerance. These are the mechanisms operating out in the body tissues.
Precisely. These are the checks and balances designed to silence or eliminate those escaped self-reactive lymphocytes after they've left the central organs.
How do we keep these escape cells quiet? What's the first mechanism?
A major one is anergy, which basically means functional unresponsiveness.
So the cell's live, but it can't react.
Exactly. Think about how T cells normally get activated. They need two signals. Signal one is seeing their specific antigen presented on an MHC molecule, but that's not enough. They also need signal two, which is a costimulatory signal, usually provided by molecules like D7-1 or B7-2, on the surface of an activated antigen-presenting cell, APC.
Okay, the two-key system.
Right. Now imagine a self-reactive T cell encounters its self-antigen out in the tissues, but it's presented by a normal, resting tissue cell that does not express those B7 costimulatory molecules.
So it gets signal 1, but not signal 2.
Correct. In the absence of signal 2, instead of becoming activated, the T cell enters a state of anergy. It recognizes its target, but is functionally paralyzed and unable to mount an attack.
It's effectively switched off.
Switched off, exactly. And related to this, there are also critical inhibitory receptors on T cells that act like brakes. The two most important ones highlighted are CTLA-4 and PD-1.
Ah, CTLA-4 and PD-1. These names pop up a lot in modern medicine, especially cancer treatment.
They absolutely do, and it's directly relevant here. These receptors, CTLA-4 and PD-1, are co-inhibitors. When they bind their ligands on other cells, they deliver a powerful stop signal to the T cell, actively shutting down activation even if signal 1 and signal 2 are present.
They're the override switches.
They're crucial breaks, and we know this because, again, look what happens when they don't work. Inherited mutations in the CTLA-4 gene cause severe systemic inflammatory diseases in humans.
Proving its role as a brake.
Undeniably. And conversely, think about cancer immunotherapy. The revolutionary checkpoint inhibitor drugs are antibodies designed specifically to block the CTLA-4 or PD-1.
Right. Therapies like ipilimumab or pembrolizumab, they take the brakes off the T cells.
Exactly. By blocking these inhibitory receptors, you unleash the T cells to attack the tumor cells more effectively. It's been life-saving for many cancer patients.
But there's a side effect profile, isn't there?
There is, and it's incredibly revealing. A common side effect of checkpoint inhibitors is the development of autoimmune reactions, inflammation in the colon, skin, endocrine glands. Basically, the treatment can induce autoimmunity.
So by blocking the brakes to fight cancer, we sometimes cause the very problem those brakes normally prevent.
Precisely. It's a therapeutic paradox that provides stunning confirmation of how essential CTLA-4 and PD-1 are for maintaining peripheral tolerance and preventing self-attack in healthy individuals.
Okay, so energy and inhibitory receptors are key. What's the next major peripheral mechanism? You mentioned peacekeepers.
Yes, the regulatory T cells, or TREGs. These are absolutely fundamental to maintaining immune balance.
What defines a TREG?
They are a specific subset of CD4 plus T cells. Their key characteristic is high expression of a surface marker called CD25, and crucially, the internal transcription factor FOXP3.
FOXP3 seems critical.
It's the master regulator for TREGs. FOXP3 is absolutely required, not just for their suppressive function, but for their development and survival. If you think of TREGs as the immune system's police force, FOXP3 is their rulebook and badge.
And what happens if there's a problem with FOXP3?
Again, human genetics tells the story. Loss-of-function mutations in the FOXP3 gene cause a devastating syndrome called IPEX.
IPEX
Stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. It's characterized by severe multisystem autoimmunity starting almost from birth.
showing just how vital these TREGs are for keeping things under control systemically.
Yeah.
How do they actually suppress other immune cells?
They have several mechanisms, but a major one is secreting powerful anti-inflammatory cytokines, specifically IL-10 and TGF-beta. These dampen down the activity of other effector T cells and APCs.
And TREGs aren't just for preventing autoimmunity against self, are they? The source mentioned pregnancy.
That's a fascinating point. TREGs are essential for maternal tolerance of the fetus during pregnancy. Remember, the fetus expresses proteins inherited from the father, which are technically foreign to the mother's immune system.
So without TREGs, the mother's immune system might attack the fetus.
It's thought that TREGs play a critical role in preventing that rejection, allowing the pregnancy to proceed. They induce tolerance to those paternal antigens.
Amazing.
Mm-hmm.
Okay, so we have energy, inhibitory receptors, and TREGs. Is there a final mechanism for dealing with escaped self-reactive cells in the periphery?
Yes, the ultimate fallback. Deletion by apoptosis. Just like in the thymus, self-reactive cells that become activated in the periphery can be eliminated.
How does that happen out in the tissues?
There are a couple of main pathways. One involves internal stress signals within the cell activating the mitochondrial pathway of apoptosis, often involving a pro-apoptotic protein called BIM.
Okay.
But perhaps the more famous pathway involves cell surface interactions, specifically the Fas receptor and its Fas ligand,
A death receptor pathway.
Exactly. When lymphocytes are repeatedly stimulated or perhaps recognize self-antigen under certain conditions, they can upregulate the Fas receptor on their surface. Other cells, often other activated T cells, express Fas ligand.
So Fas ligand binds to Fas.
Yes, and that engagement is a direct instruction. It triggers a cascade inside the Fas-expressing cell, leading directly to its death by apoptosis. It's a mechanism for deleting chronically activated or potentially dangerous cells.
And if this Fas-Faz pathway is broken,
Mutations in either the FAS gene or the FASL gene lead to another human autoimmune disease called the autoimmune lymphoproliferative syndrome, or ALPS.
what does ALPS look like?
Patients with ALPS have massive accumulation of lymphocytes, enlarged lymph nodes and spleen, and develop autoimmune reactions, particularly against blood cells. It often resembles lupus. It proves how vital this death pathway is for controlling lymphocyte numbers and preventing autoimmunity.
We should also quickly mention immune privilege sites. That's another way tolerance is maintained, right? By hiding antigens.
Sort of, yeah. Sites like the testes, the eye, and the brain have mechanisms that limit immune access and responses. Antigens in these areas are somewhat sequestered.
But what happens if there's trauma?
That's the risk. If trauma breaches that privilege and releases those previously hidden antigens, the immune system might see them for the first time and mount a strong inflammatory response. Post-traumatic uveitis, inflammation in the eye after injury is an example.
Okay, so we have a really complex system of central and peripheral tolerance designed to prevent self-attack. But clearly, it fails. We know autoimmunity results from a mix of susceptibility genes and environmental triggers.
That's the current understanding, yes. It's rarely just one thing. Most autoimmune diseases are complex multigenic disorders.
How do we know it's multigenic?
Twin studies are key. If you look at identical twins who share 100% of their genes, the concordance rate for developing the same autoimmune disease is significantly higher than for fraternal twins who only share about 50% of their genes.
That points to genetics.
It does. But crucially, the concordance rate for identical twins is almost never 100%. If one twin has lupus, the other identical twin doesn't automatically get it.
Meaning genes aren't the whole story. There must be an environmental component, too.
Exactly. Genes load the gun, but environment often pulls the trigger.
Let's talk about the genes first. Which ones have the biggest impact? The HLA genes seem paramount.
Absolutely. The HLA complex, which encodes the MHC molecules that present antigens to T cells, contributes the largest chunk of genetic risk for most autoimmune diseases. The way your specific HLA molecules present self-peptides can make you more or less susceptible.
Is there a really strong example of an HLA association?
The classic one cited is the link between ankylosing spondylitis, that inflammatory disease of the spine, and the HLA-B27 allele.
The risk increase there is phenomenal, right? Like 100 to 200 times higher?
It's one of the strongest HLA associations known in medicine. It's a massive increase in relative risk.
But does everyone with HLA-B27 get the disease?
No, and that's the critical point. The vast majority of individuals who are HLA-B27 positive never develop ankylosing spondylitis. So B27 confers strong susceptibility, but other genetic or environmental factors are clearly required. It sets the stage. It doesn't guarantee the outcome.
Okay. Beyond the HLA genes, recent research has uncovered other important non-MHC genes. The source highlighted PTPN22.
Yes, PTPN22 is probably the most frequently implicated non-HLA gene across a range of common autoimmune diseases: rheumatoid arthritis, type 1 diabetes, lupus.
What does PTPN22 actually do? It's a phosphatase.
It encodes a protein tyrosine phosphatase called LIP. Think of it like a crucial off switch or a damper for lymphocyte activation signals. Tyrosine kinases generally promote activation, while phosphatases like LIP reverse that, applying the brakes.
And the PTPN22 variants associated with autoimmunity.
They are thought to result in a version of the LIP enzyme that is less effective at putting on the brakes.
So the lymphocytes are hyper-responsive, the off switch is faulty.
That's the idea. It lowers the threshold for T cell and maybe B cell activation, making them more easily triggered, potentially impairing negative selection and tolerance, and just generally leading to excessive immune responses.
Fascinating. The other key non-MHC gene mentioned was NOD2, linked to Crohn's disease. That brings in that gut microbiome connection.
It certainly does. NOD2 is an intracellular sensor, primarily in immune cells and gut lining cells, that detects components of bacterial cell walls. It's part of the innate immune system's surveillance system.
And certain variants are linked to Crohn's, which is an inflammatory bowel disease.
Yeah.
What's the connection thought to be?
We live with trillions of commensal bacteria in our gut, and normally our immune system tolerates them. One leading hypothesis is that the NOD2 variants associated with Crohn's lead to a defective response to these gut microbes.
So instead of ignoring the friendly bacteria,
The immune system might react inappropriately, maybe failing to contain them properly or mounting an excessive inflammatory response against them, leading to the chronic gut inflammation characteristic of Crohn's disease. It's a breakdown of tolerance to our own microbial residence.
okay, so we have the genetic susceptibility laid out by HLA, PDPN22, NOD2, and likely many others. Now, what about those environmental triggers that pull the trigger? Infections seem key, particularly through molecular mimicry.
Molecular mimicry is a really compelling mechanism. It's the idea that a component of an infectious microbe, say a bacterial or viral protein, happens to look very similar structurally to a self-protein.
So the immune system mounts a response against the infection.
A perfectly appropriate, strong response. But because of that structural similarity, the T cells or antibodies generated against the microbe accidentally cross-react with the self-tissue that looks like it.
What's the clearest example of mimicry causing disease?
The textbook case is rheumatic heart disease. Following infection with certain strains of Streptococcus bacteria, strep throat, antibodies generated against a strep protein called M protein can cross-react with proteins in the heart muscle and valves.
Leading to heart damage after the infection is gone.
Exactly. The immune system clears the strep, but the misguided attack on the heart continues.
But infections don't necessarily need to mimic self to trigger autoimmunity, right? Just the inflammation they cause can be enough.
Absolutely. This is sometimes called bystander activation, and it relates back to that co-stimulation idea.
How so?
An infection causes tissue damage and inflammation. That inflammation activates local antigen-presenting cells, APCs, causing them to upregulate those crucial co-stimulatory molecules like B7.
Okay, the APCs get activated by the infection.
Right. Now imagine a self-reactive T cell, one that escaped central tolerance and maybe was previously anergic because it only saw self-antigen without co-stimulation, encounters its self-antigen being presented by one of these now activated B7-expressing APCs.
Oh, now it gets signal 1, self-antigen, and signal 2, B7.
Exactly. The infection-induced inflammation provides the missing signal 2. The energy is broken, the self-reactive T cell gets fully activated and starts attacking the self-tissue. The infection essentially lowers the bar for activating self-reactive cells that were already present.
So the infection acts like a general alarm that wakes up dormant trouble.
That's a good way to think about it. It provides the context, the inflammatory milieu, that allows pre-existing self-reactivity to flare up.
Before we move on, we have to touch on the striking gender bias. Why are so many autoimmune diseases, like lupus, so much more common in women?
It's a really profound observation. For SLE, the female-to-male ratio during childbearing years is about 9:1. For Sjogren's, it's similar. The reasons aren't fully worked out, but it's thought to involve a combination of factors.
Like hormones.
Sex hormones, particularly estrogens, definitely play a role. They are known to modulate immune responses, sometimes enhancing B cell activation and antibody production.
And genetics beyond hormones, maybe the X chromosome.
Yes, genes on the X chromosome are also implicated. Since females have 2 X chromosomes, while males have 1 X and 1 Y, there's more potential for X-linked genes involved in immunity to contribute, perhaps through mechanisms like skewed X inactivation. It's an active area of research, but the hormonal and X chromosome influences are considered key.
And one final point on the general mechanisms. Why do these diseases tend to be chronic and progressive? The source mentions epitope spreading. What's that?
Epitope spreading is a really important concept for understanding the chronicity and often worsening nature of autoimmune disease. It describes how the immune response can broaden over time.
How does it broaden?
Okay, imagine the initial autoimmune attack targets one specific part of a self-protein that's the initial epitope. The inflammation and tissue damage caused by that initial attack can then expose other parts of the same protein or even completely different proteins from the damaged tissue that were previously hidden from the immune system.
Creating new targets.
Exactly. These newly exposed self-components act as new epitopes. The immune system then mounts new responses against these additional targets. So, an attack that started very focused can gradually spread to involve multiple components of the tissue.
It's like throwing fuel on the fire. The damage itself generates more things to attack.
Precisely. It creates a vicious cycle, an internal amplification loop, where tissue damage releases new antigens, activating new lymphocyte clones, causing more damage, releasing more antigens, and so on. This helps explain why the diseases often persist and can become more severe over time.
Okay, that general framework is incredibly helpful. Let's now zoom in on the disease that really exemplifies systemic autoimmunity, systemic lupus erythematosus, SLE.
Right, lupus. It's often considered the prototype because it's so complex and can affect so many different parts of the body.
You described it as heterogeneous, chronic, remitting, and relapsing. What defines it pathologically?
At its core, SLE is defined by the production of a huge array of autoantibodies, especially those targeting components of the cell nucleus, the antinuclear antibodies, ANAs. These antibodies form immune complexes that deposit in tissues, causing inflammation and damage pretty much anywhere: skin, joints, kidneys, blood cells, nervous system.
And the epidemiology is quite striking.
Very much so. It predominantly affects women, especially during their childbearing years, with that 9.1 female-to-male ratio we mentioned. Prevalence is around 1 in 700 for women in that age group, and it's also significantly more common in non-white populations, particularly African Americans, Hispanics, and Asians.
Let's talk about those hallmark ANAs. Which ones are most important for diagnosis or prognosis?
Well, finding ANAs in general is sensitive. Most people with SLE have them, but not specific. Many other conditions can cause ANAs, but there are two specific ANAs that are much more strongly linked to SLE.
Which are?
First, antibodies against double-stranded DNA, anti-dsDNA. High levels of anti-dsDNA are quite specific for SLE and are particularly associated with kidney involvement lupus nephritis. Monitoring their levels can sometimes help track disease activity.
Okay, anti-dsDNA is a big one. What's the other?
Antibodies against the Smith antigen, anti-antimum. Anti-M antibodies are found in maybe 20–30% of SLE patients, but they are incredibly specific. Finding anti-SM is considered virtually diagnostic for SLE.
So anti-dsDNA is linked to severity, kidneys, and anti-enzym is highly specific for the diagnosis itself.
That's a good summary, yes.
Clinicians often use immunofluorescence to detect ANAs, and they report different patterns. What do those patterns tell us, like homogeneous, rim, speckled?
Right, the pattern reflects where the antibodies are binding within the nucleus, which gives clues about the specific target antigen.
So a homogeneous or diffuse pattern.
That usually means antibodies are binding broadly to chromatin, histones, maybe dsDNA basically, the main substance of the nucleus.
What about a rim or peripheral pattern?
That often indicates antibodies binding strongly to the nuclear envelope or peripheral chromatin, and it's frequently associated with anti-dsDNA antibodies.
And the speckled pattern?
Speckles is the most common pattern seen, but it's also the least specific. It means antibodies are targeting various non-DNA nuclear components like ISM, ribonuclear proteins, RMPs like SSARO, SSBLA, and others. You need further tests to identify the specific target.
Okay, and there's a nucleolar pattern too.
Yes, that indicates antibodies targeting structures within the nucleolus like RNA polymerase or fibrillary. It's less common in SLE, maybe more associated with scleroderma, but it can occur.
Beyond these ANAs targeting nuclear stuff, a significant chunk of lupus patients, maybe 30–40%, also develop antiphospholipid antibodies. And this leads to a major paradox.
A huge clinical paradox. These antibodies are directed against plasma proteins like beta-2 glycoprotein I that happen to be bound to phospholipid surfaces.
Okay, and they interfere with lab tests.
Yes, in standard in vitro coagulation tests, like the APTT, these antibodies interfere with the phospholipid reagents used, making it look like the patient's blood takes longer to clot. This led to the really misleading historical term lupus anticoagulant.
Misleading because in the patient,
it's the complete opposite. These antibodies actually create a hypercoagulable state. They make the blood more prone to clotting, not less.
how do they cause clotting?
The mechanisms are complex, but it involves the antibodies activating endothelial cells, lining blood vessels, activating platelets, and possibly interfering with natural anticoagulant pathways. The net result is an increased risk of thrombosis blood clots in both arteries and veins.
And this causes serious problems.
Very serious. Recurrent clots, strokes, heart attacks, pulmonary emboli, and also, critically, recurrent miscarriages because of clots forming in the placental vessels.
This whole picture, the antibodies plus the clinical events like clotting or miscarriages, is recognized as its own syndrome.
Yes, it's called the antiphospholipid antibody syndrome, APS. It can occur on its own or very commonly secondary to SLE. It's a major cause of morbidity and mortality.
Okay, let's try to pull these pieces together. The source material described a kind of pathogenic cycle for SLE, figure 6.25. How does this destructive loop get started and keep going?
It's thought to start with some kind of trigger, often an environmental one like UV radiation from sunlight, maybe certain drugs or infections. These triggers cause increased apoptosis, cell death.
So more cells are dying than usual.
Right. Step 2 seems to be a key genetic predisposition in many patients, a defect in clearing these apoptotic bodies and their nuclear material efficiently.
So the cellular debris, especially the nuclear debris, hangs around longer.
Exactly. You get this increased burden of nuclear antigens floating around. Step 3. Individuals with SLE have those underlying defects in self-tolerance. We discussed the failure of central and peripheral mechanisms. So their self-reactive B cells and T cells are available to see this nuclear debris.
And they get activated and start making ANAs.
Correct. Now comes the crucial amplification loop. These autoantibodies bind to the nuclear antigens, forming immune complexes. B cells and dendritic cells then internalize these immune complexes.
Why is that internalization step so important?
Because inside those cells, the nucleic acid components, DNA or RNA, within the immune complex engage specific toll-like receptors, TLRs, particularly TLR7 and TLR9, which are normally designed to detect viral nucleic acids.
Ah, so the immune system gets tricked. It thinks its own nuclear waste is a virus.
Essentially, yes. This TLR engagement provides a powerful danger signal, a potent second signal, that massively stimulates the B cells to produce even more high-affinity autoantibodies. And critically, it stimulates dendritic cells.
What did the dendritic cells do then?
They start pumping out large amounts of type I interferons, like interferon alpha, which are potent pro-inflammatory cytokines normally used to fight viruses.
And these interferons just fuel the fire.
They pour fuel on the fire. Type I interferons further enhance the activation of B cells and T cells, promote more autoantibody production, and contribute to tissue damage. It creates this vicious, self-sustaining cycle. UV triggers apoptosis. Defective clearance leaves nuclear junk. Tolerance failure allows ANA production to immune complexes form. TLRs get activated, interferons get made, more immune activation, more ANAs, more damage. And round it goes.
That really paints a picture of how it becomes chronic.
Mm-hmm.
And most of the damage in SLE is caused by these immune complexes depositing in tissues, making it a type 3 hypersensitivity.
Predominantly, yes. The deposition of these antigen-antibody complexes in blood vessel walls, kidney glomeruli, joints, skin that triggers complement activation and recruits inflammatory cells, leading to vasculitis and tissue injury.
Let's focus on the kidney damage lupus nephritis. You said it's common and serious.
It is. Clinically significant kidney involvement occurs in up to half of SLE patients and is a major determinant of prognosis. There's a classification system with 6 classes based on biopsy findings.
Which class is most common and severe?
Class 4, diffuse lupus nephritis, is generally the most common severe form. Diffuse means it affects most of the glomeruli in the kidney biopsy.
What does it look like under the microscope?
Class 4 is characterized by widespread proliferation of cells within the glomeruli endothelial, mesangial, epithelial cells. But the hallmark lesion, the really defining feature, is the presence of extensive subendothelial immune complex deposits.
Deposits underneath the lining cells of the capillaries.
Exactly. These thick deposits, when stained and viewed with light microscopy, often create dense, rigid-looking capillary loops that pathologists call wire loop lesions. They are very characteristic of severe lupus nephritis.
Okay. Moving to the skin, the classic butterfly rash on the face is very well known. What's happening there microscopically?
Histologically, in the skin lesions, you typically see inflammation around blood vessels and appendages, and critically, damage at the junction between the epidermis and the dermis specifically, vacuolar alteration of the basal cell layer.
Immunofluorescence is important here too.
Very important. Immunofluorescence staining of a skin biopsy, even from seemingly uninvolved skin in many patients, shows granular deposits of immunoglobulin, IgG, IgM, IgA, and complement components right along the dermoepidermal junction. This lupus band test reflects the systemic immune complex deposition.
Finally, the heart can be affected in a specific way in SLE. You mentioned Liebman-Sachs.
Yes, Liebman-Sachs endocarditis. This is a characteristic, though less common now with modern therapy, a form of non-bacterial verrucous warty endocarditis.
What's unique about it?
You get these small, sterile vegetations, typically made of fibrin, immune complexes, and inflammatory cells. They can occur on any heart valve, but classically they can be found on both sides of the valve leaflet's atrial and ventricular surfaces, as well as the valve edges. That helps distinguish them from, say, rheumatic or infective vegetations.
And besides the valves, is there a broader cardiovascular risk in SLE?
Absolutely. Patients with SLE have significantly accelerated atherosclerosis. They are at much higher risk for heart attacks and strokes, even at younger ages than the general population. Managing cardiovascular risk factors is a critical part of long-term SLE care.
Okay, that deep dive into lupus really highlights the systemic nature and the complex interplay of antibodies, interferons, and immune complexes. Let's broaden out again to related systemic diseases and advancements. We touched on the CTLA-4 PD-1 checkpoint inhibitors in cancer.
Right, and how their success, paradoxically, reinforces their role in preventing autoimmunity. By blocking these natural breaks to unleash an anti-tumor response, we often see autoimmune side effects. It's a direct demonstration of tolerance mechanisms in action.
It really connects basic immunology to cutting-edge therapy. Now let's shift to another major systemic autoimmune disease. Sjogren's syndrome. This seems focused on glands.
That's the defining feature, yes. Sjogren's is characterized by immune-mediated destruction of the exocrine glands, primarily the lacrimal glands producing tears, and the salivary glands producing saliva.
Leading to the classic symptoms.
Exactly. Intense dry eyes, which is called keratoconjunctivitis sicca, and severe dry mouth, xerostomia. Patients might also have dryness in other areas, like the nose, throat, or skin. It can significantly impact quality of life.
Is it primarily T cells or antibodies driving the damage?
It's thought to be primarily a T-cell mediated process, with activated CD4 plus T cells infiltrating the glands and causing damage, but B cells are also clearly involved.
Are there specific antibodies associated with Sjogren's?
Yes, very much so. Antibodies against two specific ribonucleoprotein antigens, SSA, Rho, and SSB, LA, are highly characteristic. They are found in up to 90% of patients with Sjogren's.
Can these antibodies appear in other diseases too?
Yes, anti-Rho SSA is also commonly found in SLE and sometimes associated with specific features like neonatal lupus if the mother has the antibody. But in the context of prominent dry eyes and dry mouth, anti-Rho and anti-LA are strongly suggestive of Sjogren's.
Beyond the dryness symptoms, which are significant, is there a more serious long-term risk associated with that chronic inflammation in the glands?
Unfortunately, yes. This is a really important point. The persistent lymphocytic infiltration and chronic B-cell activation within the salivary glands creates a perfect storm for malignant transformation.
Meaning cancer.
Yes. Patients with Sjogren's syndrome have a substantially increased risk estimated at up to 40 times higher than the general population of developing B-cell lymphomas, particularly a type called multilymphoma, mucosa-associated lymphoid tissue lymphoma, arising within the salivary glands. Ongoing surveillance is important.
Wow, 40-fold.
Yeah.
That's a stark reminder of the dangers of chronic inflammation.
Okay.
Next related disease, systemic sclerosis, also known as scleroderma. The name suggests hardening.
Literally hard skin, yes. Scleroderma is defined by three core features: chronic inflammation, widespread damage to small blood vessels, and, most prominently, progressive fibrosis, excessive deposition of collagen and extracellular matrix in the skin, and multiple internal organs: lungs, heart, kidneys, GI tract.
What drives this fibrosis? What's the underlying pathogenesis?
It's still not fully understood, but the model involves some unknown initial trigger, maybe environmental, in a genetically susceptible person. This leads to autoimmunity-activated CD4 plus T cells releasing cytokines, autoantibody production, and also direct vascular damage.
And how do those lead to the fibrosis?
The activated immune cells and damaged endothelial cells release a flood of growth factors and cytokines like TGF-beta, PDGF, IL-13 that stimulate fibroblasts, the cells responsible for making collagen. These fibroblasts become overactive and churn out massive amounts of collagen and matrix, leading to the characteristic hardening and thickening of tissues.
Is scleroderma one uniform disease, or are there subtypes?
There are two main clinical patterns based on the extent and progression of skin involvement. Diffuse cutaneous scleroderma involves widespread skin thickening, often including the trunk and proximal limbs. tends to progress more rapidly, and has a higher risk of early, significant internal organ involvement, especially lung fibrosis and kidney disease.
And the other type.
Limited cutaneous scleroderma. Here, the skin thickening is typically restricted to the fingers, hands, face, and lower arms legs, sparing the trunk. It generally progresses more slowly. This form is often associated with a specific cluster of features known as the CREST syndrome.
CREST, that's an acronym.
Yes. Calcinosis, calcium deposits in the skin, Raynaud phenomenon, episodic vasospasm in finger stows, esophageal dysmotility, difficulty swallowing due to fibrosis, sclerodactyly, thickening of skin on fingers, and telangiectasias, dilated small blood vessels on the skin.
Do autoantibodies help distinguish these subtypes or predict prognosis?
They are actually quite helpful prognostically. There are two major, largely mutually exclusive antibodies. Antibodies against DNA topoisomerase, also called anti-cell 70, are highly specific for scleroderma and are strongly associated with the diffuse cutaneous form and an increased risk of severe interstitial lung disease, pulmonary fibrosis.
And the antibody associated with the limited CREST form.
That's the anti-centromere antibody. Finding this antibody is strongly linked to limited cutaneous scleroderma and the CREST syndrome features. While patients with anti-centromere can still develop serious complications like pulmonary arterial hypertension, they generally have a somewhat better prognosis regarding rapidly progressive fibrosis compared to those with anti-CL70.
Okay, that's very useful clinically.
Okay.
Our final related condition is one that's gained recognition more recently, IG4-related disease. IG4RD. This sounds different.
It is, and it's a really fascinating and evolving area. IgG4-RD is now recognized as a distinct systemic condition characterized by fibroinflammation, a mix of fibrosis and dense inflammatory infiltrates that can affect almost any organ system.
Which organs are typically involved?
Common sites include the pancreas, causing autoimmune pancreatitis, the biliary tree, cholangitis, salivary and lacrimal glands, mimicking Sjogren's, kidneys, lungs, aorta, retroperitoneum. The list is long. Often patients have multi-organ involvement.
What are the pathological hallmarks? What defines it on a biopsy?
There are three key features pathologists look for. First, a dense inflammatory infiltrate packed with lymphocytes and, crucially, abundant IgG4-producing plasma cells. Second, a characteristic pattern of fibrosis, often described as storiform, cartwheel-like. And third, often obliterative phlebitis, inflammation blocking small veins. Elevated serum IgG4 levels are also common, but not universally present or required for diagnosis.
Is it definitely autoimmune? Do we know the target antigen?
That's the tricky part. While it clearly involves the immune system, particularly B cells and plasma cells producing IgG4, the exact trigger and the specific autoantigen, if any, remain largely unknown. It's debated whether the IgG4 antibody itself is directly pathogenic or just a marker of the underlying inflammatory process, driven perhaps by specific T-cell subsets.
The therapies targeting B cells seem to work.
Yes, and this provides strong evidence for the central role of B cells. Treatments that deplete B cells, like rituximab, have shown remarkable efficacy in inducing remission in IgG4D. This suggests that even without knowing the precise target, interrupting B-cell activity and the production of IgG4, and potentially other factors, is key to controlling the disease.
A very interesting, relatively newly defined condition. Okay, we've covered a vast amount of territory today.
We certainly have, from the basic mechanisms of tolerance failure to the complexities of specific diseases like lupus and its relatives.
To wrap this up, what are the absolute core takeaways for our listener trying to grasp this challenging topic of autoimmunity?
I think the first is that autoimmunity isn't just a switch flipping, it's a failure of multiple layers of regulation. Central tolerance, deletion via ARE editing, provides the first screen, and peripheral tolerance, anergy via CTLA4-PD1, suppression by TREGs via FOXP3, deletion via FAS-FASL, provides the crucial backup. Both have to fail.
And the actual development of disease requires that interaction between genetic predisposition and environmental hits.
Absolutely. Susceptibility genes, especially HLA alleles, but also non-HLA genes like PTPN, CONG2, or NOD2, create the underlying risk. But triggers like infections, causing mimicry or bystander activation, or UV light, are often needed to initiate the process in a susceptible individual.
And the way the disease manifests the pathology often depends on the main mechanism. Immune complexes driving vasculitis and nephritis and SLE versus T-cell-driven inflammation and fibrosis causing gland destruction in Sjogren's or skin hardening and scleroderma.
Exactly. The downstream effectors, antibodies, immune complexes, specific T-cell subsets dictate the clinical picture.
This brings us full circle to that puzzling statistic we started with, the rising incidence of autoimmune diseases, especially in developed nations where infectious diseases are much better controlled. It seems counterintuitive.
It does, because we just discussed how infections can trigger autoimmunity. But there's another side to that coin, often discussed in the context of the hygiene hypothesis.
The idea that being too clean might be a problem.
Sort of. The hypothesis suggests that reduced exposure to certain microbes and parasites early in life, due to improved sanitation and hygiene, might lead to an improperly educated or regulated immune system.
How might that connect to autoimmunity?
While some infections might actually be protective, perhaps regular exposure to certain microbes helps maintain a robust population of regulatory T cells, TREGs, keeping the immune system in balance. Without that constant low-level stimulation and training, the regulatory arm might weaken, making the system more prone to overreacting when it does encounter a trigger, including self-antigens.
So it raises this really provocative question for you, the listener, to think about. If infections have historically been triggers, why are autoimmune diseases increasing as we get cleaner? Could our modern hyperhygienic environment be inadvertently disrupting the delicate balance needed for immune tolerance? What are your thoughts on this trade-off between sanitation and immune regulation?
Are we perhaps creating an immune system that's naive and poorly regulated, making it more susceptible to catastrophic failure when challenged? It's a question with huge implications for public health and how we approach disease prevention in the modern world.
A fascinating and slightly unsettling thought to end on. Thank you so much for guiding us through this incredibly complex but vital area of immunology today.
It was my pleasure. It's a field that's constantly evolving, and understanding these mechanisms is key.